If you have olive skin, the kind that tans without burning, the kind that holds a warm golden undertone even in the dead of winter, geneticists have spent the last several years quietly rewriting where that trait actually comes from. For a long time, olive skin was treated as a Mediterranean story, a southern European inheritance, something people associated with sun-soaked coastlines in Spain, Italy, and Greece. But ancient DNA, pulled from skeletons that are thousands of years old and sequenced using techniques that did not exist even 15 years ago, has revealed something stranger and older than a coastline story. The genetic threads that produce olive skin do not begin in Europe. They begin in the highlands of Western Asia, in mountain valleys most people have never heard of, carried by a population that farmed the land there long before the pyramids, before writing, before almost anything we think of as ancient history. And once you understand what that ancient DNA actually shows, populations that seem to have nothing in common, separated by thousands of miles, different languages, different religions, different histories, start to make a strange kind of sense together, connected not by culture or geography, but by a shared thread running through their genomes that almost nobody outside a small circle of population geneticists has been paying close attention to. Before we get into what the DNA reveals, if you find this kind of deep genetic history interesting, take a second to subscribe to the channel. We put out videos like this regularly, and we try to go further into the actual science than most channels bother to. Hit subscribe, turn on notifications, and let's get into what the evidence actually says. To understand where olive skin comes from, you first have to understand what skin color actually is at a biological level, because most people think of it as a simple line running from light to dark, and that is not how it works. Human skin gets its color from two types of a pigment called melanin. The first is eumelanin, which produces brown and black tones. The second is pheomelanin, which produces yellow and red tones. Every person on Earth produces both. What differs between individuals and between populations is the ratio between the two, the total amount produced, and how that melanin is packaged and distributed inside skin cells. Skin color is not controlled by one gene. It is what geneticists call a polygenic trait, shaped by dozens of genes working together, including ones called MC1R, TYR, OCA2, IRF4, SLC45A2, and one that turns out to matter more than almost any other in this story, a gene called SLC24A5. Olive skin specifically describes an intermediate tone, moderate eumelanin, reduced pheomelanin, with a particular warm, faintly golden or greenish undertone. It is a real describable phenotype, but it is not a single genetic switch, and there is no one gene a lab can point to and say, this is the olive skin gene. It is the product of a specific combination of variants across several of these genes layered on top of one another, and unlike a lot of the more clean-cut genetic traits used in popular science explainers, this particular combination has a traceable and genuinely surprising history running back through some of the oldest ancient genomes ever sequenced. That history starts in a place called Ganj Dareh, a Neolithic site in the Zagros Mountains of western Iran, where researchers sequenced the genome of a woman who lived roughly 10,000 years ago. Her community was raising goats and beginning to farm at almost the exact same time. Farming was independently emerging in the Fertile Crescent to the west. When her genome was analyzed, researchers found she carried at least one copy of the derived version of SLC24A5, the same gene variant strongly associated with lighter, more depigmented skin in modern populations. But she did not carry the derived version of SLC45A2, a second major pigmentation gene that, when present alongside SLC24A5, pushes skin tone toward the very pale end of the spectrum common in much of modern Europe. In other words, her genetic profile was already, 10,000 years ago, sitting in that specific intermediate zone, lighter than a fully dark ancestral phenotype, but nowhere close to the palest end of the human range. That is close to a genetic definition of an olive-leaning tone, present in the highlands of Iran at the very dawn of agriculture. What makes this even more interesting is that the derived SLC24A5 variant was not unique to her community. Researchers found it was already present at meaningful frequency in both the Neolithic farmers of the Zagros and in a separate related population known as the Caucasus hunter-gatherers, who lived just to the north in the mountains of modern Georgia and Armenia. That means the variant was circulating in this general highland region of Western Asia before these two groups had even fully split apart from one another, likely tens of thousands of years earlier than the Neolithic period itself. This region, running from the Caucasus down through the Zagros range and across the Iranian plateau, was not adopting agriculture and then developing this pigmentation profile as some kind of package deal. The genetic groundwork had already been laid, deep in the Ice Age, by hunter-gatherer populations living in these highlands, long before anyone in the region planted a single seed. That underlying layer became far clearer in 2015, when researchers sequenced two ancient genomes recovered directly from the Caucasus itself, one from Satserblia Cave, dated to roughly 13,000 years ago, and one from the Caucasus Coal Day rock shelter, dated to roughly 9,700 years ago, both in western Georgia. The two genomes revealed a previously undocumented ancestral lineage, now referred to as the Caucasus hunter-gatherers, that had split away from the Western European hunter-gatherer line around 45,000 years ago and diverged from the ancestors of the Neolithic farmers a further 20,000 years after that. Crucially, these Caucasus hunter-gatherer genomes already carried the derived SLC24A5 variant, the same lighter pigmentation variant later found in the Zagros farmer woman at Ganj Dareh. That pushes the presence of this variant in the highland region back to well before the last ice age had even ended, deep into a period when agriculture was not even a distant idea yet. The same research found that this Caucasus hunter-gatherer ancestry went on to become one of the two major genetic components of the Yamnaya, the Bronze Age steppe population behind much of the later expansion into both Europe and South Asia, and one of the researchers involved in the discovery noted that this specific ancestral component is the closest match found so far for a genetic signal that runs right across the modern populations of India. This is also where the story stops being just about one isolated valley in Iran, because the Zagros farmers were genetically distinct from the other major Neolithic population of the region, the Anatolian farmers, who lived in what is now Turkey and who were the group most responsible for bringing agriculture into Europe. The two farming populations, Anatolian and Iranian, or Zagros-related, diverged from each other and developed along separate genetic lines for thousands of years before either began expanding outward. That distinction matters because it means there were effectively two separate ancient engines of expansion radiating out of this part of the world, carrying overlapping but not identical genetic signatures. And both of them, it turns out, carried variants tied to the same intermediate pigmentation profile. When the Anatolian farmers began moving into Europe roughly 8,000 to 9,000 years ago, they brought the derived SLC24A5 variant with them, and over succeeding centuries, mixed with the Mesolithic hunter-gatherers already living across the continent, a detailed genome-wide study of this admixture process found something remarkable. When researchers scanned the genomes of Neolithic-era Europeans for evidence of selection, the single region of the genome showing the strongest excess of Neolithic farmer ancestry, more than any other region tested, was the area surrounding SLC24A5. The hunter-gatherer version of that gene was being actively pushed out and replaced by the farmer version at a rate far higher than random chance would predict. This means the light to intermediate pigmentation genetics carried by these incoming farmers were not just present in the mix, they were being actively favored, spreading through early European populations faster than almost anything else in their DNA. But Europe was only the western half of this expansion. To the east, the Zagros-related Iranian farming populations were undergoing their own migrations, moving across the Iranian plateau and into the highlands of what is now Afghanistan, Pakistan, and the borderlands of Central Asia. This eastward movement is where the story connects to one of the most significant ancient DNA findings of the last decade, the genetic reconstruction of the population behind the Indus Valley Civilization, the Bronze Age culture that built the great cities of Harappa and Mohenjo-Daro. When researchers sequenced ancient genomes connected to that civilization, including a landmark sample from the site of Rakhigarhi in modern India, they found a population built from a mixture of two main ancestral sources, one related to these ancient Iranian farming populations, and one representing a deep, older South Asian hunter-gatherer lineage. Across the broader cline of individuals connected to the Indus Valley world, researchers found the Iranian-related component range from roughly 45 to as high as 80% of total ancestry, depending on the specific population sampled, with the remainder coming from that older, indigenous South Asian lineage. There is an important nuance here that is easy to flatten into a simpler story than it actually is. The Iranian-related ancestry found in these Indus Valley populations was not simply the Zagros farmers themselves picking up and moving east wholesale. Genetic analysis shows this South Asian-related lineage actually split off from the Western Iranian farming populations at a surprisingly early date, before agriculture had even been invented in either region. That means the population that eventually contributed to the Indus Valley world was a separate, long-diverged branch of the same broader Western Asian family tree, not a late-arriving colony of Zagros villagers. Still, because both branches descended from the same deep highland ancestry where these pigmentation variants had already taken hold, the intermediate, olive-leaning tone tied to that lineage rode along with both branches of the expansion, one heading west into Anatolia in Europe, the other heading east into the Iranian plateau, Afghanistan, and ultimately the Indus Valley. This matters beyond just the specific question of skin tone, because it pushes back on a version of ancient history that treated the Iranian plateau as a secondary region, a corridor people passed through on their way to somewhere else, rather than a source of major population movements in its own right. For much of the 20th century, the dominant narrative of the ancient world centered Mesopotamia, Egypt, and later Europe. with the Iranian highlands mentioned mostly as a backdrop. The genetic evidence gathered over the last 15 years complicates that picture considerably. The Zagros and the wider Iranian plateau were not a waiting room between more important centers. They were an independent source of agriculture, an independent center of population growth, and an independent origin point for migrations that reshaped the genetic makeup of an enormous stretch of Eurasia, from the Balkans to the Indus. The pigmentation variants discussed here are, in a very literal sense, one of the traceable fingerprints that migration left behind. The scale of that Eastern legacy is hard to overstate. Researchers working on this population history have estimated that somewhere between 2/3 and 3/4 of the ancestry of all modern South Asians traces back to this same broad ancestral population connected to the Indus Valley world. That is why, when you look at the gradient of skin tones across the modern populations of Iran, Pakistan, Afghanistan, and northern India, you see something that roughly tracks the proportion of this ancient Iranian-related ancestry each population carries, fading and blending as you move further from the highland core and mixing more heavily with older, deeply rooted South Asian ancestry as you move south and east. The picture is not finished yet, because there is a third major population that carried these same highland genetics into an entirely different direction. And that is the group known as the steppe pastoralists, the Bronze Age populations of the Pontic-Caspian steppe associated with the early spread of Indo-European languages. These steppe groups are often talked about as though they were a single uniform population, but genetically they were a blend formed from a mix of Eastern European hunter-gatherers and a substantial component from the Caucasus hunter-gatherers, that same highland population related, though not identical, to the Zagros farmers who carried the early version of the SLC24A5 variant. When these steppe-descended groups expanded, moving west into Europe and east across Central Asia roughly 5,000 years ago, they carried fragments of this same highland genetic inheritance with them, layering a second wave of related pigmentation ancestry on top of the one already spreading from the Anatolian farmers. This is part of why modern genetic maps of pigmentation-associated variants show such a broad, continuous footprint stretching from the Iberian Peninsula in the west to the Tarim Basin in the east, and from the Caucasus down through the Arabian Peninsula. It was never one single migration. It was multiple overlapping ones, all drawing from a shared highland source population in Western Asia. Because of this long, unbroken presence in the region, the modern populations of the Caucasus, including Georgians, Armenians, and Azerbaijanis, along with a number of smaller ethnic groups scattered through the North Caucasus Mountains, carry unusually high levels of this ancestral Caucasus hunter-gatherer component even today, often more concentrated than in populations further away that only received it through the diluted, several times removed inheritance of the steppe migrations. This is part of why researchers studying the deep roots of this pigmentation lineage frequently turn to modern Caucasus populations, alongside the ancient genomes themselves, as something close to a living reference point for what this highland ancestral genetic package looked like before it was carried outward and diluted across a much wider area. It is also worth pausing on the ancient farming population of the Levant, the region covering modern Lebanon, Syria, and Israel-Palestine, because it forms a third separate lineage in this story rather than a simple extension of the Zagros or Caucasus populations. Genetic analysis has found that these early Levantine farmers do not share the same specific ancestral alleles as the early Zagros farmers or the Caucasus hunter-gatherers, despite the relatively short geographic distance between the regions. Levantine populations, both ancient and modern, absorb genetic input from multiple neighboring lineages over thousands of years, including contributions connected to Anatolia, to the Arabian Peninsula, and to later movements originating further east. That layered history is part of why the modern populations of Lebanon, Syria, and the wider Levant show such a wide internal range of skin tone even within a single extended family, rather than one uniform inherited type. This is also why modern populations across an enormous and culturally unrelated stretch of territory, from southern Italy to Lebanon, from Armenia to northern Pakistan, from the Caucasus to parts of Central Asia, so often share this same intermediate, warm-toned skin phenotype, even though their languages, religions, and recorded histories diverge completely. It is not coincidence, and it is not simply about latitude and sun exposure, although sun exposure absolutely still plays a role in how visible the trait becomes. It reflects a shared, ancient genetic inheritance, diluted and reshaped differently in each population by thousands of years of separate history, but tracing back through overlapping lines to the same general highland zone. It is worth being careful here, because it would be easy to overstate this and turn it into a tidier story than the evidence supports. Olive-toned skin is not found only in populations descended from this Western Asian expansion. There are populations in parts of East Africa, Southeast Asia, and the Americas whose skin tone falls into a similar visual range. produced through combinations of pigmentation variants that developed separately in those populations. A pattern biologists call convergent evolution. Different genes, shaped by different regional selection pressures over different timescales, can arrive at a visually similar result without sharing the same ancestral source. This is an important distinction, because it means skin tone alone is never enough to draw a genetic conclusion about someone's ancestry. Only detailed DNA analysis, looking at the specific variants and the ancient haplotype patterns around them, can actually tell you whether two populations with olive skin got there through the same ancient migration or arrived independently. This is one of the genuinely important lessons that ancient DNA research keeps reinforcing across nearly every trait it touches. A visible feature, a skin tone, a hair texture, an eye color, can look essentially the same from the outside while resting on completely different genetic foundations underneath, built by entirely separate evolutionary paths that never had any contact with one another. The only way to tell those two situations apart is to actually sequence the genome and look at the specific variant and the DNA surrounding it, rather than relying on how a population happens to look. Separate research into the broader genetics of pigmentation and tanning response adds another layer to this picture. A study out of King's College London identified around 20 genes associated specifically with how skin responds to ultraviolet exposure and tanning, roughly half of which were already known pigmentation genes, and half of which had never previously been linked to skin color at all. Genes like IRF4, originally studied for its role in immune signaling, turned out to also strongly influence how skin tans and how much pigment is deposited in response to sun exposure. This tanning response layer sits on top of baseline pigmentation genetics like SLC24A5 and SLC45A2, which is part of why two people with similar underlying ancestry can still show visibly different skin tones depending on how their tanning response genes are configured and how much sun exposure they have actually had over a lifetime. It is also why the same underlying ancestral genetics can look noticeably different across a single family. Two siblings can carry an identical baseline pigmentation profile inherited from the same parents and still end up looking meaningfully different from one another by adulthood, simply because one of them spent childhood summers outdoors and the other did not, or because a slightly different combination of tanning response variants shifted how efficiently their skin responds to the same amount of ultraviolet exposure. Genetics sets the range a person's skin tone can occupy. Environment and lifetime sun exposure decide where within that range they actually end up. There is also a deeper evolutionary logic to why an intermediate, olive-leaning tone would have been favored in populations living at moderate latitudes in the highlands of Western Asia in the first place. Skin pigmentation across human history has been shaped by a balancing act between two competing pressures. Darker skin, rich in eumelanin, protects against the breakdown of folate, a vitamin essential for cell division, which ultraviolet radiation can degrade. Lighter skin, by contrast, allows more ultraviolet light to penetrate and trigger the skin's own production of vitamin D, which becomes harder to synthesize as UV exposure drops. Populations living at the kind of moderate mixed sun latitudes found across the Caucasus, the Zagros, and the Iranian plateau were under selection pressure from both directions at once, and an intermediate olive-leaning pigmentation profile represents something close to a genetic compromise between those two demands. That is very likely part of why this particular pigmentation combination stabilized in this specific highland region in the first place, tens of thousands of years before agriculture gave it a set of migrations to ride on. What makes all of this land differently than a simple factoid is what it means for how people think about their own ancestry. Someone with olive skin who grew up assuming their family history was a purely Southern European story may be carrying a genetic thread that traces back through the Caucasus and the Zagros Mountains to hunter-gatherers who lived there during the last ice age. Someone with olive skin who grew up in a South Asian or Middle Eastern context may be carrying that same thread, shaped instead by the eastward migrations that helped build the population base of the Indus Valley world. Neither is more or less connected to that ancient highland population than the other. Both are downstream of the same deep genetic history, reshaped over thousands of years by different migrations, different mixtures, and different local histories, but rooted in the same original place. None of this is meant to flatten the real cultural, linguistic, and historical differences between an Armenian family, an Italian family, a Pakistani family, and a Lebanese family into a single story. Those differences are real and they matter, and no amount of shared ancient ancestry changes the fact that these are distinct peoples with distinct histories, languages, and identities that deserve to be understood on their own terms. What the DNA adds is a layer underneath all of that, a reminder that some of the physical traits people take for granted, the exact tone of their own skin, can be traced with real scientific precision back through specific ancient populations, specific highland valleys, and specific moments in the deep human past, long before any of the modern identities built on top of that ancestry existed at all. It does not explain who these populations are today. The research in this field is moving quickly. Every year brings newly sequenced ancient genomes from sites across Iran, the Caucasus, Central Asia, and South Asia, and each one adds more resolution to a picture that is still being filled in. Some of the specific percentages and population boundaries described here will likely be refined as more ancient samples are analyzed, and that is exactly how this kind of research is supposed to work. But the core finding, that the genetic roots of olive-toned skin trace back to the highlands of Western Asia rather than to the Mediterranean coastlines it has long been associated with, is now solidly supported by multiple independent ancient DNA studies, and it is one of the more striking examples of how a physical trait millions of people carry every day can be read like a historical record written in the genome of migrations that took place thousands of years before anyone was writing anything down at all. It is also worth saying plainly that a field moving this fast will keep producing corrections to itself, and that is a feature of good science, not a weakness in it. Ancient DNA research barely existed as a functioning discipline 20 years ago. The techniques used to extract and sequence usable genetic material from 10,000-year-old bone were still being developed within the last decade and a half, and the sample sizes behind some of the findings described here, especially from South Asia and Central Asia, remain smaller than researchers would like. As more remains are excavated, better preserved, and sequenced with improving technology, some of the specific numbers here will shift, some population boundaries will get redrawn, and some of what currently looks like a clean two-branch story will probably turn out to have more branches once the sampling gaps are filled in. That is not a reason to distrust the core finding. It is simply how a genuinely young and rapidly maturing scientific field is supposed to behave. If you found this valuable, take a moment to like the video, leave a comment about where your own family history traces back to, and share it with someone who would find this as interesting as you did. Subscribe to the channel for more deep dives into ancient DNA, ancestry, and the genetic threads connecting populations across the world. Thank you for watching, and we will see you in the next one.